This is part 2 of our conversation about the immune system. In this lecture, we're going to talk about the adaptive immune response and briefly discuss some common diseases. Like the innate immune system, the adaptive immune system can react to diverse pathogens and has the capacity to distinguish self from non-self. However, it also has two additional features not present in the innate immune system: specificity and memory. By specificity, we mean that the adaptive immune system produces an array of molecules, each of which has the potential to target a specific pathogen it's never before encountered. The quality of memory describes the ability of the adaptive immune system to remember past infections and mount a stronger response on re-exposure. While many cells and tissues play a role in adaptive immunity, two types of cells are particularly important: T lymphocytes, or T cells, and B lymphocytes, or B cells. T cells mature in the thymus, an organ of the immune system located just behind the sternum or breastbone. B cells mature in the bone marrow of mammals. Both B and T cells circulate in blood and lymphatic vessels. They can also be found in the spleen, liver, and lymph nodes. One of the hallmarks of adaptive immunity is the ability to target specific pathogens. The specificity of the adaptive immune system is in part the result of antibodies produced by B cells. An antibody is a large protein that carries sugar molecules attached to some amino acids. An antibody binds to foreign molecules that occur naturally on or in microorganisms and participate in normal cellular functions for those microorganisms. Such a molecule is termed an antigen, which is a molecule that binds to and leads to the production of antibodies. Antibodies can be found either on the surface of B cells or free in the blood or tissues. Collectively, antibodies make up a significant portion of the proteins present in the blood. Antibodies have a distinctive structure that reflects their specificity for a given antigen. Here, the simplest antibody molecule is a Y-shaped protein made up of 4 polypeptide chains, 2 identical light chains, shown in blue, and 2 identical heavy chains, shown in green. These 2 chain types are so called because of their relative molecular weights. Covalent disulfide bonds hold the 4 chains together. The light and heavy chains are further subdivided into variable and constant regions. Within the variable region of the L and H chains are regions that are even more variable, called hypervariable regions. Together, these hypervariable regions interact in a specific manner with a portion of the antigen called the epitope. Each of the billions of different antibodies has a distinct set of hypervariable regions that recognizes a unique epitope. Binding of antibodies to antigens is the first step in recognition and removal of microorganisms. Binding alone is sometimes enough to disable a microorganism. In these cases, binding can lead to precipitation of the antibody-antigen complex. More often, the microorganism is destroyed by a different component of the immune system, such as a phagocyte or the complement system. Antibodies are members of a family of proteins with common structural features. As a group, they are called immunoglobulins, or Ig for short. There are 5 classes of immunoglobulin: IgG, IgM, IgA, IgD, and IgE, each with a different function. These classes are defined by their heavy chains, which differ in the amino acid sequences of their constant regions. There are also 2 types of light chain. These 2 types of light chain occur in all 5 classes of antibody, but any given antibody contains only 1 type of light chain. Immunoglobulin G is by far the most abundant of the 5 antibody classes. This antibody circulates in the blood and is particularly effective against bacteria and viruses. In addition, immunoglobulin G is the only class of antibody that can cross the placenta because of the presence of receptors on placental cells for a portion of this molecule. The ability of IgG to cross the placenta provides protection to the developing fetus. Immunoglobulin M is typically a pentamer. It is particularly important in the early response to infection and is very efficient in activating the complement system and stimulating an immune response. Immunoglobulin A is usually a dimer consisting of two antibody molecules linked by a joining chain. It is the major antibody on mucosal surfaces, such as those of the respiratory, gastrointestinal, and urinary tracts. It helps to protect mucous membranes from infection. It is also present in secretions, including tears, saliva, and breast milk. Immunoglobulins D and E, like immunoglobulin G, are monomers. IgD is typically found on the surface of B cells. This immunoglobulin helps initiate inflammation. IgE plays a central role in allergies, asthma, and other immediate hypersensitivity reactions, which are characterized by heightened or an inappropriate immune response to common antigens. The 5 classes of antibody are also called isotypes. B cells can actually change the class of antibody they make, a process known as isotype or class switching. A process called clonal selection is the basis for antibody specificity. Every individual has a very large pool of B cells, each with a different antibody on its cell surface. Collectively, these antibodies recognize a great diversity of antigens, but each individual antibody can recognize either only one antigen or a few antigens. An antigen from a foreign cell interacts with the B cell, or a small set of B cells, that has the best fit between the antigen and the membrane-bound antibody. Binding leads the B cell to divide and differentiate into 2 types of cells. One type, plasma cells, secrete antibodies having the same specificity as the parent cell. The second type, memory cells, are long-lived cells that contain membrane-bound antibodies having the same specificity as the parent cell. Clonal selection explains immunological memory. The first encounter with an antigen leads to a primary response. In this response, there is a short lag before antibody is produced. The lag is the time required for B cells to divide and form plasma cells. The plasma cells secrete antibodies, typically immunoglobulin M. The level of IgM in the blood increases, peaks, and then declines. A secondary response takes place if there is re-exposure to the same antigen, even months or years after the first exposure. The secondary response is quicker, stronger, and longer than the primary response. During this response, the antibodies are released by memory B cells. These cells respond more quickly to antigen exposure than naive B cells. In addition, the pool of memory B cells is larger than the pool of naive B cells for a given antigen, explaining why the secondary response produces more antibodies for a longer period of time. Vaccines take advantage of this secondary response and provide future protection from an infection. In vaccines, an antigen from a pathogen is deliberately given to a patient to induce a primary response, but not the disease outcome. So how do we end up with such a diversity in B cells? As a B cell differentiates, different gene segments are joined in a process called genomic rearrangement that produces a specific antibody. For heavy chains, shown to the left, there are 4 key types of gene segments. These segment types are variable, or V for shorthand, diversity, or D, joining, or J, and constant, or C. During B cell differentiation, the DNA in this heavy region undergoes recombination so that just one of these segments makes part of a functional heavy chain gene, and the intervening DNA is deleted. The assembled VDJ segment encodes the variable portion, and the C segment encodes the constant region. Light chain genes, shown to the right, also contain multiple copies of gene segments, except they do not have a D gene segment. The VJ segment encodes the variable portion, and the C segment encodes the constant region. The result of this process is that each B cell encodes a single heavy chain and a single light chain, and each B cell expresses a unique antibody. We generally think of DNA as being a stable blueprint present in identical copies in all of the cells in our bodies. B cells are an exception. As a result of genomic rearrangement, the DNA in each mature B cell is different from the DNA in every other mature B cell, and different from the DNA in other cells in the body. B cells and their antibodies have limitations. For one, B cells on their own can make antibodies only against some antigens. For other antigens, they require the assistance of other cells. 2, B cells are sometimes ineffective against pathogens that take up residence inside a cell, as do the bacteria that cause tuberculosis. To handle these and other kinds of pathogens, there's another part of the adaptive immune system which depends on the second type of lymphocyte, the T cell. T cells do not secrete antibodies. Instead, they participate in cell-mediated immunity where cells, not antibodies, recognize and act against the pathogens. There are two major subpopulations of T cells, helper T cells and cytotoxic T cells. Helper T cells help other cells of the immune system by secreting cytokines. One of their key roles is to activate B cells to secrete antibodies. They also activate macrophages, cytotoxic T cells, and other cells of the immune system. Cytotoxic T cells can kill other cells. They are activated by cytokines released from helper T cells. They are particularly effective against altered host cells, such as those infected with a virus or that have become cancerous. These two types of T cell are differentiated by the presence of different glycoproteins on their surface, CD4 on helper T cells and CD8 on cytotoxic T cells. The ratio of these two classes of T cells can be used to assess immune function. All T cells, like B cells, originate in the mammalian bone marrow. Unlike B cells, T cells mature in the thymus. A mature T cell is characterized by the presence on the plasma membrane of a T cell receptor, or TCR, a protein receptor that recognizes and binds to the antigen. TCRs recognize antigens with a specific structure. There is a great diversity of TCRs that differentiate from one another, but each T cell has just one type of TCR on its surface. Binding of TCR to an antigen triggers the T cell to divide into clones, resulting in a pool of T cells that are each specific for a given antigen. As is the case with antibodies, the diversity of TCRs among different T cells results from genomic rearrangement of V, D, J, and C gene segments. TCRs are different from antibodies in important ways. For one, they are not secreted like antibodies, but are always membrane-bound on the T cell surface. Another difference is that the T cell receptor does not recognize an antigen by itself, but instead recognizes an antigen in association with proteins that appear on the surface of most mammalian cells. These proteins are encoded by the major histocompatibility complex, or MHC. The MHC is a cluster of genes present in all mammals that encodes proteins on the surface of cells. It is composed of many genes with a high rate of polymorphism, meaning that there is a lot of variability in the gene sequence, and consequently the protein sequence, among individuals. In humans and mice, these genes are divided into three classes. When an antigen enters the immune system, it may be recognized by an antibody directly or be taken up by antigen-presenting cells, the macrophages, dendritic cells, and B cells. An antigen-presenting cell takes up the antigen and returns portions of it to the cell surface, bound to an MHC class II protein. Helper T cells recognize processed antigen along with MHC class II molecules by their T cell receptors. When a receptor binds to an antigen in the MHC class II protein, the helper T cell releases cytokines that activate other parts of the immune system, including macrophages, B cells, and cytotoxic T cells. Cytotoxic T cells also recognize antigen displayed by host cells, but in this case, the antigen is presented in association with MHC class I molecules. Because class I molecules are present on virtually all cells, cytotoxic T cells recognize and kill any host cell that becomes abnormal in some way. For example, a virus-infected cell often expresses viral antigens and MHC class I molecules on its surface. Cytotoxic T cells recognize the antigen in MHC class I molecules and kill the cell. As another example, Tumor cells express novel antigens along with MHC class I proteins and may also be eliminated in some cases by cytotoxic T cells. Once activated, T cells divide and form clones of helper or cytotoxic T cells. Some cells of each type are memory cells that provide long-lasting immunity following an infection, as in the case of B cells. The ability to distinguish between self and non-self is acquired during T cell maturation. Of the possible T cell receptors that are generated by genomic rearrangement, only some are useful, those that react with the host's own MHC molecules. Useful T cell receptors must not react with molecules normally present in or on cells of the host. A sorting process is therefore necessary so that only some T cells mature and others are eliminated. As T cells mature in the thymus, they interact with cells of the epithelium of the thymus. Those that recognize self-MHC molecules on epithelium cells are positively selected and continue to mature. Those that react strongly to self-antigens in association with MHC are negatively selected and eliminated through cell death. The result of this sorting process is twofold. For one, T cells become MHC restricted. This means that they interact only with antigens that associate with MHC proteins. Helper T cells interact with foreign antigen plus MHC class II proteins, and cytotoxic T cells interact with foreign antigen plus MHC class I proteins. The second result of the sorting process is that T cells exhibit tolerance. That is, they do not respond to self-antigens. Those antigens present as T cells mature are labeled as self and do not elicit a response. Those not present are non-self and, if encountered, do elicit a response. The ability to distinguish self from non-self is critical. This ability is acquired during T cell maturation. Failure in this ability leads to autoimmune diseases in which the immune system becomes active against antigens of the host. Autoimmune diseases can be debilitating as T cells or antibodies attack cells and organs of the host. Let's talk about 3 common human pathogens. The flu, or influenza, is caused by an RNA virus that infects mammals and birds. It is notable for causing seasonal outbreaks or epidemics. The flu causes hundreds of thousands of deaths each year and millions of deaths during pandemics. Cells infected by viruses secrete cytokines that bring macrophages, T cells, and B cells to the site of infection. These cytokines, when produced in abundance, lead to many of the symptoms commonly associated with the flu. For example, the cytokine interleukin-1 produces fever. Part of the success of the virus lies in its ability to spread easily. Part lies in the virus's ability to evade the immune system. There are 3 major types of flu virus, called A, B, and C, and many different strains of those types. Once in the cell, the virus replicates its genome and makes more virus particles. However, replication is prone to error, so there is a high rate of mutation that leads to changes in the amino acid sequences of antigens present on the viral surface. This process is called antigenic drift. It allows a population of viruses to evolve over time and evade memory T and B cells that remember past infections. Antigenic drift leads to a gradual change of the virus over time. As shown to the right, the flu virus is also capable of sudden changes by a process known as antigenic shift. The viral genome consists of 8 linear RNA strands. If a single cell is co-infected with 2 or more different flu strains, the RNA strands can reassort to generate a new strain. Antigenic drift and shift make it difficult to predict from year to year which strains will be most prevalent and therefore what vaccine will be most effective. Like the flu, tuberculosis is spread through small droplets dispersed in the air when a person with active disease coughs or sneezes. Also like the flu, the TB pathogen replicates inside host cells. But unlike the flu, TB is caused by a bacterium, not a virus. TB results from infection with Mycobacterium tuberculosis, a bacterium with several unusual properties. It is very small with a compact genome, lacks a plasma membrane outside of the cell wall, and replicates exceedingly slowly. M. tuberculosis takes about 16 to 20 hours to divide, compared to 20 minutes for the common intestinal bacterium and model organism E. coli. TB is very common. About 1/3 of the world's population has TB. Symptoms are absent in most cases because the bacteria are in a dormant state, kept in check by the immune system. However, about 10% of cases are active, characterized by chronic cough, fever, night sweats, and the weight loss that gives TB its other name, consumption. TB primarily affects the lungs. The bacteria enter macrophages in the alveoli of the lungs where they replicate. The macrophages then release cytokines recruiting T and B cells to the site of infection. These lymphocytes surround the infected macrophages, forming a structure called a granuloma. The granuloma helps to prevent the spread of the infection and aids in killing infected cells. Many TB cases remain asymptomatic. However, the bacteria can sometimes overcome host defenses, especially if the immune system is compromised or suppressed, as in the case of co-infection with HIV. Treatment for active disease requires a long course of multiple antibiotics. TB diagnosis poses challenges, especially in these cases of asymptomatic infections. The tuberculin skin test is performed by injecting protein from the bacteria under the skin and observing whether or not the reaction in the form of a hard raised area occurs. A positive skin test is an example of a delayed hypersensitivity reaction caused by the recruitment of memory T cells to the site of inoculation. Malaria is caused by a single-celled eukaryote of the genus Plasmodium. Several species infect humans, but Plasmodium falciparum is the most common and most virulent. Malarium is both devastating and common. There are about 500 million cases and 2 million deaths every year, mostly in sub-Saharan Africa, and mostly of children. Malaria causes cyclical fevers and chills and can lead to coma and even death. The malaria parasite is transmitted to humans by the bite of an infected mosquito. The parasite thus bypasses the natural protective barrier provided by the skin. The malaria parasite spreads to the liver and then to red blood cells, where it completes its life cycle. The progeny can be taken up again by mosquitoes and passed to another host. The malaria parasite is able to evade our immune system. It resides in liver cells and red blood cells where it cannot be easily detected by macrophages, B cells, or T cells. Infected red blood cells can be removed by the spleen, but the malaria parasite has evolved a way to avoid this filtering action. In this process, the parasite expresses an adhesive protein that inserts itself on the surface of red blood cells. This protein interacts with proteins on the surface of cells lining blood vessels, helping infected cells stick to blood vessel walls and keeping them in the circulatory system and out of the spleen. The protein, PFEMP1, shorthand for Plasmodium falciparum erythrocyte membrane protein 1, could conceivably be a target for antibodies or T cell receptors. However, it is encoded by one of about 60 genes, each slightly different from the others. The parasite expresses just one of these genes at a time and can change which one is expressed in a process called antigenic variation. The great and ever-changing diversity of this protein, both in a single parasite and in the population as a whole, makes it a moving target for the immune system. Despite continuing research, there is no vaccine for malaria. Researchers hope that the sequence of the malaria genome, completed in 2002, will provide new targets for vaccine research. In the meantime, efforts to control malaria focus on measures to control mosquitoes, such as nets and insecticides, and on treatment with antimalarials. This brings to a close our discussion of immune systems.